So we started recording, and welcome everybody to the first episode of Plastic Pollution Podcast, a podcast where we talk with the researchers behind published papers, a bit in-depth view on the published research. And today with us, we have, actually, can you introduce yourself so I don't mispronounce your name?
Yeah, the same to me with yours. Okay, my name is Cristóbal Galbán. I am a Spanish researcher who works in Chile since 6 years ago, and I work in the Universidad Mayor. And we have been working in microplastics since 3 years, more or less.
Mm-hmm. Yes, and my name is Stjepan Budimir. I come from Croatia, but now I work and live in Finland. My background is also I started about 5 years ago working with microplastics, but now I'm not working in science in research. I'm in a private sector, but this was my contribution to the research effort. Yes, so your paper was titled Microplastic Ingestion Cause Intestinal Lesions in Intertidal Fish, Girella laevifrons, or did I pronounce the Latin name wrong?
It's Girella laevifrons, but it has a common name in Chile. Its name is baunco.
Baunco.
Yeah, maybe it's easier for you to pronounce.
Baunco, yeah. So I really liked your paper, so to say. I did my master thesis 5 years ago. Was about I wanted to look into microplastic in fishes in Adriatic Sea, and one of the big questions then and still is like, what does actually microplastic does to fishes? Does it harms the fishes, and in which way? So your paper is looking into that topic.
Yeah. You please. Sorry.
Yeah. So you, so to say, fed fishes with polystyrene particles.
Yeah. That's the first when we are starting to set the experiments. What we can do, what question we can answer. We say we look for the database or looking for a plastic who, in small quantities, could have a damage. We found some of them, and since we live in Chile and we are far from everywhere, we only were able to purchase this kind of plastic, and we started to design different experiments. This is the first work that we have been doing. We started with the physical damage because usually when you hear about microplastics impact on the fauna, including fish, they say that they clog the system, so the animal is satiated, so he's not able to eat. And that was a question that we never read a paper talking about. That was always we assume that if the animal eats plastic, usually he will clog their intestine. Sorry for the English. And
Yeah.
And the other thing that we have been looking for is that maybe if there is this system clogging, there should be another effects on a tissular on the tissues. So we first feed the fish. We do these experiments, and then we decided to. Well, previously we decided to look for the histology, because if you have some problem at histological point, or in the histology or in the tissues, there should be some implications for metabolism. So that was the main point of the paper. Instead of looking to the metabolism, let's look to the first barrier, which is the intestine or the gut. It depends, because some fishes have the different kinds of intestines conformation depending on their feeding. And the selection of this fish was based on a previous paper that we performed in 2017, when that was our first paper on microplastics. So, because we see that the numerous fish usually have more microplastics than other fish, because of their feeding strategy, If you are omnivorous, you don't differentiate. You eat everything you have on your view. So baunco, which is the fish that we selected, was the fish in wildlife or in nature that has the higher amount of microplastics. Also, we found a selection of color, but we are planning to start these experiments during this year. But with this coronavirus crisis and this the political situation in Chile also is not very calm. It's there is a lot of things in South America. We weren't able to start these experiments.
Yeah, it's kind of, so to say, this. I think researchers always face, so to say, numerous problems. It's only there is in every place the problems are a bit different, and
usually this is not the case.
You know what's interesting now, like for these issues, in Europe, in a lot of places, and also in Finland where I worked, and some places where I was looking to do my PhD, the problem was also with permits to work with adult fishes, So that was like one thing that I liked, that you're working with adult fishes and not just the juvenile phases. Because for me, if you would be doing just juvenile phases for some weeks, it's kind of it's a bit weird, so to say. It's not so. It's just a part of the picture.
I think so. Usually, when you want to work with wildlife and perform experiments, it's very difficult, and we understand this as a researchers. But you need to pass a lot of steps with the ethics permission. So, for example, one of the critics now is published, so we could comment about that. Coming from the reviewers of this paper was the low number of samples, and we agree with that. But when we perform the ethics committee review of our experiments, they 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 ask to us, because the first experiment was planned with more individuals, just for the statistical force of the statistical power of the experiment. So we decided to reduce it at maximum, because it will be easier to get a permission for that. For example, there is another paper on microplastics on marine mammals performed 2 years ago by Nelms in the UK, and they do some experiments with two, if I can remember, yes, 2 or 3 otarids, I think, and you think, okay, there is only 2 animals, but they found that when they feed these animals with fish with plastic, you find some plastic in the fecal pellets. So I think the ethics is one of the. I don't want to say that this something that blocks science to advance. I think we need, as researchers, we need us to think in our experiments, especially now. 20 years ago, probably the researchers didn't experience these problems. To look our science in the more in the best way for the animals that you are experimenting, obviously.
Yes, I agree with you 100%. It is very, very deep topic, and it's like this, so to say. I think everybody who went into studying biology, and especially working with environmental issues, it is because profound, basically, love towards the nature, animals, and trying to preserve it. So it is kind of that weird ethical question of how to prove something with harming at least animals as possible. So, and it is like what you said, the ethics says use as least animals as possible, and the reviewer says have as big a sample as possible. So, but I think you did, what was it? You had 30 samples.
Yeah.
Okay, so I mean it's okay.
For each treatment, we planned to do 50 samples per each treatment, but they did not allow us, and we understand this issue, so we reduced at maximum. But was one of the main points of the paper and the main critics that resides from the reviewers, which are welcome always.
Yes, so say hi to all the reviewers, so anybody who is listening. So let's go through then forward with your work. So you took you caught wild caught adult fishes, and you went through this process of acclimatization, and then we come to the microplastic exposure. So you had a control group, group A, and group B, B being with a substantially higher amount of microplastics. So the spiked sample was used polystyrene. So, and I've seen quite a few papers using polystyrene. And why exactly did you choose polystyrene?
We choose polystyrene because we know that polystyrene, especially polystyrene, it's a polymer, okay? But when it degrades physically, it degrades into dimers, trimers, and monomers of styrene that we know that they are toxic. We searched for this in the Institutional for Cancer Research. I think it's an institution of France who performs an assessment of the cancer capability of some compounds. They assess and they say, okay, this compound it's able to produce this kind of cancer or this kind of cancer or another kind of cancer based in the previous publication from different researchers, and that was of the main point. We were looking for a plastic that usually is able to do some kind of effect that we know, but we try to, because we were looking for not only the tissular damage, but maybe we were planning to analyze if there is some levels of monomers or dimers in the muscle of the fish, in the edible tissue. So this part is not finished yet. We started the analysis 1 month ago, but we cannot be able to start the analysis because we decide to close the laboratory for this time. But I think the idea was to test if this compound that maybe is not very toxic, maybe I say because we don't know, we have some evidence that it provokes cancer, but if we find these monomers, dimers, or trimers, or polystyrene in the tissue, it will be another interesting result because we are talking usually that when you eat some fish that has plastic, we don't eat the gut of the fish. We eat only the edible part of the fish, which is the meat. Not the meat, but you know what I'm saying. And
the muscle tissue.
Yes. And the thing is that we are not exposed to the microplastics as consumers, but we are exposed probably to this bunch of chemicals that this plastic is transferring to the fish. I think there is evidence from, for example, organic compounds, typical organic compounds like PAHs or polybrominated diphenyl ethers or other compounds that are transferred actively from the gut to the muscle and the edible tissue. So maybe we could find this, but from the part of the microplastics per se, from their composition.
Yeah. That was basically, you kind of answered my, so to say, already next question. I mean, I saw from your previous work, you've been working with organics pollutants. You have experience with
we are
that.
working on that. Now I have two research lines. One is microplastics, which is an interesting line, and also the environmental behavior is similar to persistent organic pollutants in a some way, because if you look to the definition of persistent organic pollutants, they are not degradable, they are from the wildlife, they are harmful for the wildlife, they are able to be transported by physical mediums like the water, the air, and also the they are organic, so, and they are man-produced, so they have a lot of similarities. So I wasn't scared to start to working with that, and I think a
Yeah,
lot
it
of
makes
researchers
sense.
from the area are moving between these two worlds finally.
Yeah, it is like when I started, kind of like I went with the microplastic in fishes because I had this, so to say, interest in fish ecology, and then from that side. But I understand that at one point I really hit some wall when it comes to experience work with the organic pollutants. So I think it's a perfect. I mean, it's a great combination of skills. So that's really good. Yeah. So I'm just kind of going through the paper so I don't forget some of my notes. Overall, the paper is quite, so to say, very understandable. It's kind of very to the point. I liked it. So we are now here. Yeah, this food preparation. So I kind of didn't understood. How did you? Could you maybe say like what exactly have you done here? Did you use normal fish food, so to say, and then break it, or how did you
No,
do that?
no. What maybe in the paper is very not well clear. It's not very clear. We try, but usually we cook our own food, taking into account the metabolism necessities of the fish. We look for another paper we cite in that publication, and we look for what is the amount of protein that the fish need and the vitamins that the fish need, and we use one agglutinant. I don't know the word in English. Is to mix all together and to stay and to have a solid phase. We use agar, which is not very. It's a low energetic food. So, and in that food we used the typical food of this kind of fish. In terms of protein, we use mussel and concholepas, with this typical mollusk from Chile, and it's widespread along the coast. And these fish are present a preference for this fish. And also we included spirulina, which is a typical macroalga that provides all the vitamins that the fish need, because we were not able to isolate the microalgae or the algae to feed the fish from the field. I think this could be a weakness of the paper, but we try to maintain the caloric support, and to, especially for the control fishes, to maintain their performance. And also, in the case of the other fishes, we made the same mixture, but we include increasing quantities of plastic. But this is not very easy to do. We do a lot of tests to ensure that our food was enough quality for maintain the fish. And this is another thing that was also commented to us, because it's very difficult, because there is not much information about about that kind of fish. We have information from similar fishes from other areas of the world. For example, there is another fish called from the same genus, which is Girella nigra, which is well established in the United States, in the Atlantic, I think also in the Pacific coast. But this is this is one of the difficult parts, because you need to be very meticulous, or you have to put a lot of caution when you do this kind of experiments. Because if you use commercial food, within that you are changing the preference performance for commercial feed, and for commercial feeding, sorry. And this is quite an important issue for this kind of experiments. For example, there are a lot of papers that they use, for example, that was the thing that we thought about our paper, that we don't use the typical zebra fish or the medaka fish. I think the point of the paper is that we use a real fish not real, but you understand this from nature. And that's I think that's the main point. But the part of the feeding and the food cooking was one of the most critical parts in this kind of experiments.
Yes, thank you for so to say clearing that. I was so to say questioning these things while reading your paper. Well, this is one of the reasons why I started this, because there is, I think in every paper there is something that is either hard or didn't fit in the last version. So it's great that you explained this thoroughly. But yeah, I think you put it quite so to say quite well. So. Yes, and yeah, you had the control group then so it was that potential food effect was in the control group. So yeah, okay, thank you for clearing that. And then, so to say, we are going to. Then you had the histological examination of the fish tissue, of the intestine tissue. So this. How to put it? This is not, so to say. It's maybe a bit harder to put it in so to say easy words. But yeah, basically, you had this so to say 4 steps, or zero being no changes, and then slight, moderate, and severe changes towards the tissue. So. I think we shouldn't maybe talk too much about it. This is maybe need to be just read. Or did you wanted to say something about this part?
About how we scale the tissular damage. I think we are talking about this.
Yes, please do.
Was not very easy, because the usually where what you do, and the previous experience, and in the paper we had an anatomical pathologist, which is very expensive with animals, not with fish, but with he's a veterinary scientist, very recognized in Chile and known for this. Also, he works with human tissues, Carlos Gonzalez, and he helped us, because he told, okay, we have this scale which usually is a little bit subjective, because it depends of the eyes of the viewer. You need to have experience on that, and you need to have trained on that. It's because we started to work with him, because he's an expert on that, and we also see this kind of analysis in another paper. So we see why we cannot apply this. This will be not easy, but it's in our hands to do that. Carlos Gonzalez has his office near to me, so I think it occurs naturally, the collaboration. And when we talk about that, he says, okay, I will review the samples, and by your way, one of your team members review the samples using this scale. And we use three different people. And when we were looking to the results, they were very, very similar among them. And when the team members, which are not experienced, put in their Excel, okay, about the samples in the report, he says, "I'm not sure about that. What do you think?" And Carlos review another time the samples that the students were in that, but not the students, the team members say, "I think I see that." But then it was reviewed by an expert, because this kind of not quantitative, because it's qualitative, the thing that you do, it's usually very important to have an expert, because you need to have a clinical eye. I don't know if you understand the expression, but
yeah.
And that was one of the interesting parts of the paper, because usually when you look for papers on histology, they are very visual, most of them. So you don't have we have one here, we have two here. No, it appears that the tissue is damaged because you have the structure here is broken, and these have been desquamated. So in our scale, I think this is, and usually it's interesting to have 3 or 4 people working on that from a different point of view, because usually when we work in the laboratory, and if you have work in the laboratory, and probably all of our listeners, says that it's very important that you are doing one step in your experimental design or in your experimental work. It's very important that the same person takes the whole step, just to eliminate the probable variability that will appear from one person to another, which is not the same if you are doing some sums or additions, doing math, that you say 2 plus 2 is 4, and everybody is able to do that. But when you are talking in this kind of a special world of histology, that you need to have a clinic eye, I think it's very important to have this in mind.
Yes, it is this kind of like we are, is it called that visual or terror? I mean, some of the things, and so to say, minimizing. You can never eliminate the human factor, but you can minimize. And
Yeah,
yeah,
that's right.
and you sound like you took this challenge quite seriously. So that's very good.
Try this, because I think I'm not an expert in histology. I come from another area. So I think that one of my weakness would be this part. So I try to take this in a serious way, because probably if we didn't take this in a serious way, probably we'll have some problems later with the publication of the paper, or also not only with the publication, because publication is the final step, and this for me is not very important. Also, we were not sure about if we answer the question that we are having, and that we question to us.
Mm-hmm. Yeah, I understand what you're saying, I think. Okay, so should we move on to results?
Okay, looking to our results, the main thing, the first thing that we wanted to examine if is if the particles could be found in the intestine of the fishes. So we examined, and the counting was very difficult because of the small size of the polystyrene, which we didn't thought about this, because the particles are so small that they were very difficult to count. So we decide another strategy. We do a random sampling in every intestine and different parts. And when we saw an accumulation of these transparent spheres, because they look like transparent in the microscope, we put this random sampling as positive. So it's because of that we presented our results in percentage of occurrence, but not in the number, because I think when you are trying to count, it's very difficult to count everything, especially in these sizes. So we decide with the other authors, especially with histologists, that it will be better, and maybe more important, just to show if there is occurrence or not with each treatment. And our feeling about the possible results that we could have were the reality. So we found, and it's clearly seen in the graph that we present as figure 1 in the paper, is that the control has none of these kind of spheres that we use. Then the treatment now, which samples, which the concentration of microplastics was very small. We only find plastics in a small sample size, and then when we go through the higher concentration samples, we found that there is a clear, significant higher occurrence of these particles. So for us, this was a proof of that our approach in the experimental design was good. This is our first. Probably if we did not find this occurrence, probably we will not find we'll not finish the experiment because they say, okay, if we don't find any particles, maybe if we have another thing, maybe it's not related to the exposure to particles. So later, we go through to the second part of the experiment that was looking for this tisular damage. Uh, there were there are plenty, or not plenty is not the correct word. There are a lot of indicators that you could use, but we selected these that were used previously in similar stress by pollutants, and most of them, especially the leukocyte infiltration and hyperemia, are usually related to inflammatory response, which is usually what happens when you are stressing an animal, especially with physical damage. Okay, it's the same that when you are hurt with a knife or something. The first is hyperemia, and it's because you have a lot of blood, and then is the leukocyte infiltration because the immunitary response to this to a possible infection. So uh, this what this we focus first in these things, but there are some things that we need to take into account because the intestine instead. All of us know that there is an internal organism, but uh, also the intestine has a lot of external exposure. I don't know if it could be easy to understand, but uh, you must remember that you have not an entering area, which is the mouth, and a delivering area, which is the anus. So uh, usually it's one of the most exposed to external influence in our bodies and also in the fish. For example, looking to another kind of pollutants, for example, in the case of persistent organic pollutants, the main entrance in the organism is by feeding. So it's because this is the organ that it's in contact with output, and also it's in contact with the external media. I think this is more easy to understand. And what we find is that there was a consistent increasing damage in our results, especially looking first for hyperemia, and we know that the controls have some moderate to slight damage, but you must understand at this point that this damage is the normal in the intestine. Also, intestine has usually a lot of hyperemia because it's the way of transport of nutrients from the intestinal lumen to our vessels and then to our body just to feed us and get our energy or nutrients that we need. And also in the case of fishes. But what we found is that it was an increasing of the moderate to severe when we are going when we are increasing the concentrations. Especially, it's quite interesting the results found in the case of hyperemia, which is increasing in the levels of moderate to severe. But in the case of the leukocyte infiltrations, we find a similar trend. But looking to our results, the control and the lower concentration samples are not very different because we have absent, slight, and a little bit moderate. But when we go through the most higher concentration, uh, we found that the severe increases and the moderate increases significantly. So probably the damage is much higher. Then there are another two indicators, which is the degradation per se of the tissue, which is the Billy cell loss and the Krebs cell loss. These cells are very implicated in the movements of the food through the intestine and also in the exchange of nutrients. Okay, so what we found looking to the hyperemia and leukocyte infiltration that something is happening in the level in the most concentrated with plastics food. And looking to bilious loss, we find a dramatic uh, increase in bilious loss, which means that there is a lot of physical damage, which is similar that and not similar, but it's in agreement with what we found previously with leukocyte infiltration of hyperemia. So the tissue is getting hard because of the feeding. And in the Krebs cell loss, we found a similar response, but not as heavier at the C-level cell loss. But the most important is that Billy cell loss are are more exposed to these pollutants because these kind of cells are moving per se, so they are more exposed to this kind of damage. This is the main results that we have, and also all of them were statistically significant, especially when you test the lower concentration with the higher concentration, So this is an agreement that also with what we found in the occurrence of polystyrene particles. So it seems that there is a mechanism that we are introducing plastics into the intestine of the fish, and that these plastics are doing some kind of damage in terms of immune response, looking to the leukocyte infiltration and the hyperemia, and also in the physical structure of the tissue. So the next step, looking to the results, will be to look for any genetic response or overexpression or down expression of some genes. But this will be another kind of work in the future.
Yeah, it's a very so to say it's one step in the long chain of understanding the systems. Can I ask you now at this point, like, so because there were some amount of damage in the control group.
Yeah.
So what I was thinking, like, these damage to intestines, this physical damage, and the responses, do you think could it be, so to say, how much is it because of the plastic and the, so to say, toxicity, I mean the chemicals properties of plastic compared to just physical so to say physical burden to the intestines?
I understand your question. I think that we argue in the paper, for example, in the part of discussion, that maybe the plastic is releasing some chemicals, but none of the response that we were looking for in the present paper were related to a chemical stress. So it's very difficult to differentiate if this kind of response is related to the proper physical damage or to the chemical compounds that could be present in the plastic per se. But the thing that we could argue is that looking to the controls is that usually the intestine is a highly stressed organism because of the movements and the passing of food through it, and it could be an important physical damage. I think this is quite important because when we talk to other researchers in some meetings about these results, they say, okay, but usually your intestine has always hyperemia and leukocyte infiltration in a lower grade, and is usually related only to the physical damage that it's perceiving the intestine during consumption and digestion. In the case of the chemicals, we argue in the paper, as I commented before, that maybe there could be another stress generated by this polystyrene degradation in monomers or dimers, but we were not able to prove them because we did not analyze by GCMS some samples, because we didn't thought about that when we are doing the experiments. So now, for example, and usually it's something that happens to scientists, that okay, maybe if I could have done this, the results will be very better. And but this idea came to us during the reviewing of the paper, and we have this in mind for the future experiments. But it will be interesting to find this kind of chemicals. But another thing that it's quite important is that access or to the use of these equipments usually in South America is not very easy. That was usually one of the main things that we present when we are doing science at the end of the world, is that we call Chile usually. And this is not an excuse, but now, for example, we are starting to collaborate with different researchers from different countries. So it will be quite interesting to start this kind of research, especially with some environmental chemists which are working with me now, and we were discussing about that in the future.
Yeah,
I don't know if I answered well your question. Sorry.
Yeah, you did. So what did I wanted to? Yeah, about this environment. So just wanted to stress that the difference between the first and second group was the concentration was 100 times higher in the second one than in the first one. So the first one was how to say environmentally relevant concentration, and the second one was much higher than environmentally
I think it was
relevant.
An outrageous concentration. But that is the other thing that we discuss with people with who works in microplastics, is that okay, it's important to work with environmental relevant concentrations, but taking lessons from people who study metal concentrations, usually most of the results in environmental toxicology with metal was obtained because the use of higher concentrations, because you are able to see, okay, at environmentally relevant concentrations of, for example, of tantalum, which is a strange element, okay, it's probably that you don't see any damage or any effect. But when you use increasing concentrations, you are able to find what is the tipping point for an impact or response of the animal you are testing on. So I think it's quite important to bear this in mind, because usually, for example, when I talk with researchers from other areas, especially from ecophysiology, because they usually work with temperatures, with all the range that they use are environmentally relevant at the level of at the individual level, which temperature exposure, for example, is lower, and it's relevant because we live in this range of temperatures. But in the case of plastics, it's projected that in the future concentrations will be very high. Also, if we were more rigorous about that, the people could argue, and also we argue to us in the discussion of the paper in the meetings, that probably the fishes are not only exposed to a single kind of plastics; they are exposed to a different kind of plastics, different sizes. So it's quite important to bear this in mind, that this is we use a wildlife animal, but there is an experiment that was performed in control conditions in the laboratory. So the extrapolation to the nature should be taken with care. And this is one of the things that we say in the conclusions, I think. And the other thing is that this will give you some idea or what could happening to the fishes in a plastic world in the future.
Yeah, I agree with you. I didn't mean to say something negative in this way, that there was a much higher concentration. I think I would always have a part of the experiment with much higher concentration to see the trends and so on. But one also thing that I've noticed in the environment is that when it comes to plastic and most of the things in the environment, it's not that you have uniformly distributed amounts of anything. So you will have, this kind of like a concentration points. Let's say on a beach, it's easy to see. Like you might have like 100 meters of relatively clear beach, and then in that one corner of the beach where the currents and the waves and the wind is, you will have like so much more plastic on one pile. So let's say the microplastic in the aquatic environment is not always uniformly distributed. So it probably the fishes are moving from the parts that they are more and less plastics. So it is interesting to see how does it go with this substantial increase in concentration. I found that very interesting.
Oh, thank you.
So, do you think could we kind of wrap up the paper? There are some things other that I wanted to talk a little bit, but maybe for the paper, did you want to maybe say something more? I had a feeling that we kind of rounded it up quite nicely.
I think the most important thing of this paper, being honestly, is that we were able to, in a very small laboratory with limited space and fundings. If you look to the acknowledgements, we got a lot of the researchers who are working in the paper have funding, but usually it's not very easy to get that funding. Usually we use our own funds, taking, okay, I bought something for this project, but I could use that for this project, okay? Usually laboratory staff. And this is a quite collaborative paper, I think, which is, from my point of view, one of the most important part of science now, and especially for the future, is that I am environmental chemist, for example, one ecophysiologist working with us, one histologist, and usually, for example, in the case of histologists, they are and they recognize that. Sorry.
Yeah.
They recognize that usually they work on histology only, and for Carlos, which is the histologist, it was quite interesting to start this kind of collaboration, and I think that the information that we got with this kind of backgrounds from the different researchers working on the paper increase it significantly the paper per se. This is one of the most important, and is what we are seeing in science now. That usually is more usual now to find collaborative papers than individual papers. That I'm not saying that individual papers are bad. Usually they are good also, but to increase the collaboration is an important fact that occurred in that paper, and also the results that we found were interesting, I think, for the community.
Mm-hmm. Yes. And now, when you mentioned that, do you have paper open in front of you, so to say?
Yeah.
I would like if you could list all the collaborators with their names and associations, so I don't butcher anybody's name. And it would be, it's of course, we have to give record where record say.
And also is quite very important because the first author of this paper, which her name is C.Ahrendt, when she was doing the experimental results, he's working in our life science department at the University Andrés Bello, which is my former university. Now I moved to another one, and now this work initially comes from her idea, and now she is working with a foundation which is called Plastic Oceans, and she has the affiliation of that foundation, and now she started to be one of the most important peoples in terms of relation with society in Chile about the plastic impacts on the wildlife and also on the beaches, and she will start the PhD during the next year, and we are still working together. In the case of the second author of the paper, which is J.Perez-Venegasa, he finished his PhD working with marine mammals and plastics, and he has published several papers on this point, on this topic. Also, M.Urbina is a researcher from the Universidad de Concepción and the Instituto Milenio de Oceanografía. He's quite important expert on microplastics, and also he's an ecophysiologist of fishes. And he performed a very good reviewing of the paper during the draft, and he helped us a lot during the writing of the paper. Carlos Gonzalez is the director of the veterinary science faculty at the University of Andrés Bello, and he is the expert histologist that helped us in the experimental section, and her support was very important. P.Echeveste is a researcher of the University of Antofagasta. He is an ecotoxicologist, and we worked together since years, and we are also good friends. And her views to the experimental design of the paper was also important for us, and also we think he increased the quality of the paper. Marcel Aldana is an expert on ecology of these fishes. He has been working with fish ecology and the relationship with the media during a lot of time. Now she is working in the Centro de Investigación e Innovación para el Cambio Climático from the Universidad Santo Tomás. J.Pulgar, which is the co-correspondent author of the paper, is a fish ecophysiologist. He has been working his whole life with Girella, and he knows the fish very well, and it was important for us to hear his recommendations, and also he support us during the setup of experiments with a lot of experience, because the first time that we performed the experiments, all the fish died, and was I think these things happen, and he said to us, okay, maybe we are having problems with the salt or with the salinity or with the temperature. So he was finally one of the most important parts of the paper in terms of not only in the experimental and giving us the possibility to use their laboratory, also in the discussion and the presentation of the results. And finally, it's me that I was working during this first redaction of the paper at the Departamento de Ciencias de la Vida from the Universidad Andrés Bello. But now I moved to another institution, and now I'm working in the GEMA Center for Genomics, Ecology and Environment from the Universidad Mayor. And we moved 6 months ago, but now the university is closed, the labs are closed, and everything is in boxes yet. So maybe we will start to doing new experiments I expect by the end of the year. We don't know what's going to happen with this pandemic state that we are living.
Yes. Thanks everybody. My greetings to all the co-authors, and congratulations on a nicely published paper. Thank you, Cristóbal, for being with us. Yes, and
welcome everywhere, every time you want. We are open.
Thank you. I suppose you will be continuing this work, and in general, so to say, I hope this kind of podcast that I started, it's a bit of a, so to say, a result of, well, basically like this, being at home and thinking, what can we do, and it's kind of crazy to think about, it's like we are talking across the world, exchanging ideas, and maybe I'm trying to find a good silver lining. If this didn't happen, I probably wouldn't have time to stop and think about some of the things. So it's like I'm trying to make the best out of this and to help.
For example, now, in my research group, we are thinking about just looking to what we have done and what we could do in the future. So I think it's a good time to rethink what are we doing and where are we going as researchers, as not only as researchers, because maybe the people which could be listening this podcast, there will be another kind of professionals, I think it's a good time to think and read and have family time. For example, usually I spend a lot of time in the laboratory and in the office working, and now I am spending a lot of time with the kids, which is not bad, but I think it's a good moment to think about that, because usually you only are with the kids 100% of your time, 100% of your time during holidays or weekends, but during the week and teaching the kids or trying to do homeworking, I think it's a good time to think about not only about our most most not only about your family or your people but maybe more about what is happening to this world. I think plastics is only one of the parts, but we could talk about a lot of problems that are happening in the world, like pollution in general, climate change, global change, political changes. I think this could be a good time for most of it.
Yeah, I think this is like that. Maybe thinking about how we are so connected.
Yeah.
It was like in a very brutal way reminding us that we are very very connected. But I'm trying to be optimistic that it will kind of that the lessons learned, that we will start thinking in a more positive way in the future.
Yeah, I think so.
Yeah. So with these positive thoughts, I would end this episode. So that is it for now.
Okay.
Thank you,
Thank you a
Cristóbal.
lot, and keep listening this podcast and the future podcasts.